Deep down in us, we find what makes us function, what makes us human. Our proteins. And now, we see them like we've never seen them before. The spleen is a purple bean-shaped organ located deep within the left side of our rib cage. This is where our blood is being recycled and stored, a hub for our body's core defense against diseases. But there are still mysteries to unveil about the spleen. In the Human Protein Atlas project, the proteins that build the human body are being mapped out, and the result is available on the HPA Open Access website. Now, the team is using these proteins with groundbreaking technology to visualize organs like the spleen in completely new ways.
I spend all days looking in the microscope and imagining what would this look like in 3D. Now there's a technique that allows me to look in 3D, and it becomes like a whole new world. It's a fascinating world when you see it in 3 dimensions like this.
The spleen is an important part of our immune system, always ready to protect our bodies from outside threats. It is built out of white and red pulp. The white pulp consists of immune cells.
But you can clearly see here the structure of almost like clouds or rounded balls. And these are actually part of the white pulp, the lymph follicles. You probably see part of the red pulp here through one of these follicles, and you can see where it's not as dense.
The red pulp is surrounding the white pulp. Its main task is to filter the blood and remove defective red blood cells and bacteria. The red pulp is also a storage for white blood cells, which can rapidly be mobilized to leave the spleen and assist in an immunological response.
All that is green here is smooth muscle cell. Smooth muscles in the capsule lying like wavy fibers that surround the spleen can make the whole spleen contract if it is needed for letting out white or red blood cells.
The different parts of the spleen communicate with the nervous system.
What we see is now only this very fine network of nerve fibers. These are then the communication areas where the nerves then talk to the immune system. This whole area of the communication between the nervous system and the immune system is a mystery to research. If you imagine that you can use this technique and use different colors to look at different specific cell type, you can actually start to understand which cells are involved when the immune system talks to the nervous system and vice versa. How does this crosstalk actually work? And I think that's very important.
When we explore our world in new ways, we discover new things about us, about life itself. And now we see our proteins like we've never seen them before.